Welcome to this episode. Today, we're diving into a crucial topic in early-stage neurological biotech: how contract research organizations and academic labs help confirm mechanism before programs move forward. It's a deep dive into independent validation and why it's so essential.
Yeah, it's a really important conversation, especially because early-stage biological research is inherently risky. Internal findings often don't replicate when tested externally, which can lead to costly mistakes if you're not careful.
Exactly. And that's why Biotech International Institute, or BII, has taken a structured approach. They're moving their early neurological programs into a third-party validation phase that's built around defined assay packages, independent review, and clear go or no-go decision points.
Right, and it's key to note that the goal here isn't to make clinical claims prematurely. It's about answering preclinical questions with rigor and transparency. So it's really about strengthening the science before moving on.
So let's start with the role of CROs. They're often seen as just service providers, but their contribution is much more foundational. They bring standardized execution to the table. CROs use established methods and documented workflows to generate structured, reproducible data. For BII's neurological portfolio, they handle things like receptor pharmacology confirmation, safety and off-target screening, pharmacokinetics, mechanism of action assays, analytical characterization, and in vivo replication.
So they're kind of like a scientific quality control factory, right? Ensuring every test is done consistently and thoroughly documented so that results can be trusted and compared across studies.
Exactly. And that's critical because it moves a program from, we believe this mechanism is worth exploring, to here's what independent testing shows so far. It's a shift from assumption to evidence.
But CROs aren't the whole story. Academic labs bring a different kind of value: scientific depth and mechanistic insight.
Yeah, they complement CROs by diving into the biology itself. They help refine hypotheses, evaluate models, and develop biomarker strategies. They ask questions like, is the proposed mechanism biologically plausible? Which pathways should be prioritized? What biomarkers can reliably measure biological response?
And that's a really important distinction. While CROs focus on standardized execution, academic labs provide intellectual rigor and independent scientific insight. They help understand why a platform might be biologically relevant, not just whether a single test is positive.
Right, and that independent perspective can challenge assumptions and refine the direction of research. It's a critical part of the validation ecosystem.
Now, this all ties into BII's use of assay packages and go/no-go decision gates. These assay packages are like a scientific blueprint. They define the question, the method, the materials, controls, and criteria for advancement or rescoping.
And having these packages prevents validation from becoming scattered or unfocused. It also helps control costs and clarifies the purpose of each study.
Plus, the gated model produces tangible outputs at each checkpoint: CRO reports, milestone updates, and yes/no decisions. It replaces narrative with data-driven milestones, which is essential for disciplined decision-making.
So validation isn't just about generating more data. It's about making better decisions, whether to advance, repeat, refine, pause, or redirect a program.
That's a great point. And to make this more concrete, let's look at how this applies across BII's neurological programs.
Sure, starting with Neuroforal, their lead program. It's a receptor-selective small molecule targeting neuroinflammation. They've completed inventor stage characterization and preliminary animal work. Now the focus is on independent receptor pharmacology confirmation, safety screening, and in vivo replication.
So the key questions here are, can receptor activity be independently confirmed? Are there any safety or off-target concerns? Can the in vivo findings be replicated? And does the biological profile support continued preclinical development?
CROs and academic labs have a clear role in confirming the mechanism and early profile credibility for Neuroforal.
Then there's Mycoforal, the second program. It's a neurotrophic pathway candidate aimed at neural recovery biology. The priority here is analytical confirmation before moving to broader biological validation.
So the focus is on analytical identity, structural characterization, purity, stability, assay readiness, and biomarker selection. The question is, do we fully understand the candidate before diving into complex biology studies?
That sequencing is really responsible. It avoids premature assumptions and ensures the foundation is solid.
Finally, neuro reset, which is earlier stage. It's a multi-target conjugate concept with lead candidate selection underway.
Validation questions here include chemical and analytical suitability, receptor activity, conjugate stability, and pharmacokinetics. The priority is defining the right candidate before commissioning studies.
So it's about disciplined progression. Don't rush into every study, but define the right candidate and then commission the right work.
When you step back, this whole validation approach is really about risk management and capital discipline.
Absolutely. Independent validation shows that BII isn't just relying on internal assumptions. They're managing scientific risk through defined questions, external testing, milestone checkpoints, visible risks, and go, no-go decisions.
And that's critical, because early-stage biotech can get prohibitively expensive if weak assumptions persist unchecked. Third-party validation helps identify strengths and weaknesses early, enabling responsible capital allocation.
The goal is better science, not faster or unsupported claims.
And that brings us to the bigger picture. CROs and academic labs aren't just service providers. They're partners in scientific rigor.
They help answer fundamental questions. What is real? What is uncertain? What should be tested next? What should be paused or rescoped? What data would increase confidence?
This partnership transforms mechanism hypotheses into measurable, credible evidence. For BII, third-party validation is the pathway that determines which programs advance, which need refinement, and which should be rescoped before unnecessary capital is deployed.
And that's how stronger, more responsible research stage platforms are built.
Well, that was a thorough look at how CROs and academic labs help confirm mechanism in early-stage neurological biotech. Hopefully, it gives listeners a clear sense of why independent validation is so critical.
Yeah, it's a complex process, but absolutely essential for advancing science responsibly. Thanks for joining us today.
Thanks. Talk soon.